So I'm going to talk briefly about data analysis. We actually cover data analysis more in the labs later on in the term, so I won't go into a lot of detail here. And some of this I've already sort of preluded, I guess. Excuse me, I just ran up the stairs. Catch my breath. Okay. So we've got a block design. Remember, I said the way we analyze is we basically look for brain areas whose bold response matches the predicted time course of our stimuli. So that's something like this. So for a block design, we don't expect that the brain activation is going to go vertically straight up and then be flat. It's going to have the sort of curviness of the hemodynamic response function built in. So it might look something like this, which shows the peakiness and then also the undershoot during the off periods. And so what you're seeing here at the top is our what we call our reference time series, so what we predict brain activation in task-relevant areas to look like. And then down here, we see actual time courses from different individual voxels in an fMRI study in an individual's brain. Some of these don't render super well on the stupid projector, but some of them you can see, like the bottom left and bottom middle, have pretty clear block design kind of patterns to them. And this was visual motion, moving dots versus static dots, so it's a really good driver of activation in V5, motion-sensitive cortex. And I knew where to look, so I sort of fished around and found some very clear ones. But the question is, which other ones might have something of that pattern, because always we've got some level of noise. And in a lot of experiments, you don't find any brain areas that show something that's that clear-cut, because you've got tighter contrasts or more complex kind of designs. All right, so to fit the model, we would sort of take that on-off time course and just correlate it with the time course in each part of the brain. And the result is a fitted time course, which is what you're seeing as the purple line here. And the question is, how strongly correlated is that with the original data? So this first one, the answer is not so much. I'm not sure how well you can see these. If not, you can take my word for it, and I'll post the slides. This one, much more so. And other ones you can see. So this is an interesting one. So on the top right, you can see that there's actually, so first of all, there's this sort of linear drift in the signal. We're not worried about that. But there is sort of an on-off alternation along with a fair amount of noise and that linear drift. And I can't quite read there the significance, but that one's actually significant. So you can find that even when the time course doesn't sort of hit you between the eyes as being obviously a block design, there's enough of that systematic variance in it that you'll be able to find that. And of course here I'm using a block design, which is sort of a very obvious one to look at. Of course, if you imagine those jittered event-related designs I showed you, where it just looks like, it could be any old time course, but we know it's systematically what we predict the bold response to be based on our stimuli. You wouldn't be able to eyeball that, so you'd rely on the math. I mean, you always rely on the math anyway. But again, you just see which brain area is correlated strongly with that time course. So when you see something in a paper where somebody has shown a brain and there's some bright, beautiful sort of blobs of activation on it, what they've done is this kind of correlation, right? Found those brain areas or those individual voxels that showed a systematic pattern, and then applied some statistical threshold to it to say, okay, let's see only those voxels that are above p equals 0.05 or whatever their threshold is, and those they color in blobs. One sort of important thing to keep in mind, and this is more when you're doing analysis, it's usually sort of under the hood when you're reading somebody else's paper, but the statistical values are not equivalent to the size of the activation or the bold amplitude. And the reason for that is if you have taken stats, this is hopefully somewhat familiar, so the formula for something like the t-test, which is just a difference between a pair of conditions, the formula for t is the difference between those conditions divided by the variance, right? And so the more variance you have, or the more noise in your data, the smaller your t-value will be. And so what we're trying to show over on the left side here is that we've got one condition where you've got quite a large amount of variability. And the sort of mean baseline level of activation is 4.3. The mean task-related activation is 6.3, so that's a difference of 2. And then the variance is 0.87. And so our t-value there ends up being 2.3. You don't have to worry about solving the math. It's done for you. So that's our t-value. Here, we have a much weaker signal change. So the difference between task and baseline is 2.2 versus 1.2. So it's this magnitude of 1. So it's half the size of the difference that it was here. But the variance is also much lower. And you can see that both by the signal value there, but also just looking at the time course. And so the t-value ends up being much bigger because the ratio of signal to noise is better. And so where this becomes important is when you look at brain activation maps, typically they're statistical maps. So they're plotting as the color scale, like the t-values or the z-values, whatever statistical values. And you'll see some really bright areas, those are the biggest statistical values. That doesn't necessarily mean those areas showed the strongest activation. They showed the most statistically robust or statistically significant activation. But as you can see here, those might be relatively small signal changes, but also low noise. And that can happen because the noise across the brain is going to be different. So it may seem like a subtle point, but it can be an important point if you're thinking about which areas show sort of are most sensitive to something versus which areas show the strongest brain response overall, or something like that. Okay. And then I mentioned the thresholding. And multiple comparisons comes up in virtually any brain imaging technique because we're almost always doing lots and lots of statistical comparisons compared to a lot of behavioral research where you do comparatively few. So when we're doing thresholding, we're basically saying we're only showing, I talked about those maps, we're only going to color code activation in the areas that are above our statistical threshold. The question becomes what threshold to use. Let's not worry about what to plot down there. I kind of talked about that already. But again, this issue is that in a typical fMRI image, you might have something like each slice is 64 by 64 voxels, so volume pixels, and you might have 27 slices. So if we do the math, and we're analyzing each one of those voxels independently, it's got a time course. We're looking to see if that correlates with our stimulus presentation. So with something like that resolution, we're performing well over 100,000 statistical tests. So if we say our p is 0.01, which might sound kind of conservative compared to 0.05, which is a little more standard, that means that we're guaranteed over 1,000 false positives. So over 1,000 voxels within our analysis are going to look like they're significant, but they're not. It's just random chance. And that's what you're seeing in this bottom image is just the same brain map in a case where there was no actual activation. And they analyzed it using some model of what they thought the activation could be. And at p of 0.05, you get noise all over the place, including outside the brain, which should make you very suspicious unless you believe in ESP. And as you increase the threshold to 0.01, again, conservative by sort of typical psychological standards, still pretty noisy. I mean, you've seen stuff at all is a problem, certainly outside the brain. And then at 0.001, now we're maybe a little more conservative, but still a few false positives. So the solution is some sort of multiple comparison correction. The one that they teach in stats class typically is called Bonferroni correction, which is you just divide by the number of tests you're doing to get a more conservative alpha or p-value. The problem with that is that with so many statistical tests, it ends up being overly conservative, and you end up making your blobs of activation smaller than they should be. And the principled reason why it doesn't make sense is that the Bonferroni correction is based on the assumption that every statistical test is independent of every other test. And so it makes sense to correct for the total number. But in fact, in the brain, that doesn't hold, right? So this voxel in my left, or this is my right hemisphere, right superior temporal lobe, is next to some other voxel in my right superior temporal lobe, and they're probably measuring basically the same brain activation. They're probably within the same brain area. We'd expect them to be correlated, so they're not independent. So it doesn't make sense to correct as if they're independent. And that's where it becomes practically sort of overly conservative. One approach is to mask the brain that you're analyzing with a template brain, or more simply, like in that last one, we saw stuff outside the brain. That's sort of misleading because in practice, we don't expect anything to go on outside the brain, so we don't actually analyze the data outside the brain. So right there, we're reducing the number of contrasts or statistical tests that we're doing by eliminating all those voxels that are in the image but not inside the brain. Another one is, and this is the one that really is what people use in practice most of the time, is some sort of cluster-based thresholding. So here the idea is that we consider the smoothness of the image, or what we expect to be sort of the inherent autocorrelation or correlation between adjacent voxels, and use that as a way of predicting just by chance what's the odds. Okay, so let me step back. So we run our analysis, we threshold our image at, say, 0.05, and we're likely to get some individual voxels that are lighting up, but then blobs or clusters of voxels that are all touching each other that are lighting up, which are, a priori more likely to be active brain areas because there's multiple ones next to each other, which is what we expect knowing how the brain is organized. But we can't just sort of arbitrarily say any cluster over 20 voxels in size, we'll call that significant, because that's just pulling things out of the air. So the idea here is saying, okay, if we just take the image and sort of by chance randomly, sort of pushed values into this brain image, applied this threshold and looked for clusters, what is the probability of finding a cluster of 20 voxels just by chance in a random noise image? What's the probability of finding a cluster of 100 voxels by chance? And so you can set a threshold, because everything's given there as a probability. So you can say, okay, now we're applying a 0.05 p-value threshold based on the probability of the cluster size rather than the individual voxels. And that's a much more principled way that sort of respects what we understand about how the brain is organized and is objective in terms of applying a p-value rather than just sort of coming up with some arbitrary number. It's actually a real danger because most fMRI software allows you to set the p-values, the thresholds. Some of them actually have sliders, so you can just sort of move it up and down until it sort of looks nice, and then say, oh, I like that threshold, I'm going to use that as my image. But then, of course, you have to write your results section and explain why you chose a p-value of 0.00729, and people get suspicious. So this allows you to be objective but principled about it. But even with this, there have actually been, over time, some realization that initially even the defaults in the fMRI analysis software packages were overly liberal. And there were a series of papers that were published over the last few years demonstrating this. And there was a whole uproar, and for a while, like, the popular press was like, oh, fMRI is bogus, like, all these guys, they're just grossly inflating their claims, and we can't interpret anything, which isn't quite true. But they've adjusted things, so now the software packages are a little more conservative and a little more principled about how they do it. But as you read through papers, even still sometimes you see stuff that gets published, and it is like some arbitrary number of voxels that they chose as a cluster threshold, or the p-value just sort of doesn't make sense, or something like that. So you do have to be, even as a consumer of brain imaging papers, you have to attend to what they did in terms of their statistical thresholding and think about whether that actually makes sense and what the downsides or limitations of that set of choices might be. Oh yeah, and there's one called false discovery rate, which is another variant of the cluster-based approach. Another thing people do is, rather than analyzing the whole brain, which is what I've talked about, which is often called a mass univariate analysis, univariate being one variable, so one voxel, and mass being you're doing this lots of times because there's lots of voxels. Rather than analyzing the whole brain, you might do a region of interest, or ROI analysis. The idea here is saying, okay, I have some a priori prediction that I'm doing a face study. I'm interested in the responsiveness of the fusiform face area. So I don't really care about the rest of the brain. I'm just going to draw a circle around the fusiform gyrus, or the fusiform face area, and restrict my analysis to that one area. And what that buys you is a lot of sensitivity, because now you're not correcting for 110,000 statistical tests, you're correcting for maybe 250, because that fusiform face area is quite small. And so you don't have to worry so much about the cluster-based thresholding, and you have a greater sensitivity. And at the same time, it's principled in the sense that a priori, anybody who's reading the paper can understand why you would pick that area. What's not okay is to sort of post hoc look at the brain and do your whole brain analysis, not find what you want, and then say, oh, actually, I think I'm just interested in that area. So let's reanalyze it with just that, or let's reanalyze it with these 12 regions of interest. Now, if a priori you had 12 regions of interest, that's still okay. right? If you can justify it. And the reality is that you kind of know that sometimes people did it one way and then they didn't get what they want and they went back another way. But hopefully there's still some rationale for why they chose that, because these areas were shown active in previous studies and so on. But it can give sensitivity, like if you are dealing with patients and it's hard to get enough subjects to get a really big statistically robust group, and you might be stuck with a really small group, so you might use that as a way of boosting your sensitivity, or if they're very tiny effects, or you're doing it in sort of an exploratory fashion, to say, okay, let's do this, and if we can find it, that gives us motivation to run a bigger study, something like that. So that's all still sort of around the univariate approach. An alternative is to do what's called a multivariate analysis, which basically means looking at multiple voxels at once in the fMRI context. Multivariate analysis has other meanings in other areas of psychology and statistics. But here, and this approach was really initially shown in Haxby et al. In early 2000s. And here what they were doing was, they were fMRI researchers, they'd looked at lots of these brain images, they'd done these face processing studies, seen the fusiform face area active, but they noticed that the fusiform face area wasn't the only area that lit up during face processing. And there were some areas that were consistently deactivated, so showed reduced bold signal relative to, or I guess another way of thinking of that is more bold signal during the control conditions than when seeing faces. And some people might say, oh, we don't care about that. But they're kind of like, is there any consistency to like this overall spatially distributed pattern of activation? So in this study, they presented multiple runs. So each run is like a 5-minute scan. So you can say they ran 8 of these runs. And they had some faces and some houses, other stimuli, but we'll just focus on faces and houses. So some face blocks, some house blocks, 8 different runs. And what they did is they just sort of analyzed their data, so did that boxcar kind of correlation, but didn't apply a statistical threshold. They just took everything and said, okay, some of these are really big bold responses, differences positive, some are negative, some are smaller. They did restrict it, as you can see in the color map here. They didn't look at the whole brain. They restricted it to sort of inferior temporal lobe areas involved in visual processing, but they didn't threshold it. And they just said, okay, here's the spatially distributed pattern of activation in one slice on the even numbered runs. So runs 2, 4, 6, 8 of this experiment. Here's the spatially distributed pattern of activation unthresholded in runs 1, 3, 5, 7. What's the correlation between those? So how consistent, if we look at sort of voxel by voxel, how similar are the values, whether it's positive or negative, whatever, between the odd and the even runs? And it's pretty high. So consistently, so about 0.8 correlation, which is very high correlation. But when they did that and said, okay, how do the even runs for faces correlate with the odd runs for houses, they found a negative and weak non-significant correlation. So what that's basically telling us is every time you see a face, yes, your fusiform face area lights up more strongly than it does for other things, but other brain areas are doing things that are consistent every time you see a face as well, and very different from what those brain areas are doing in distributed fashion when you see houses. So it suggests that what our brains are doing is more than just this sort of, and it sort of links up to more higher order philosophical ideas about how the brain is organized. So this area of, or the idea that one brain area does a particular thing is really what I've called neophrenology before. And through the 80s and 90s, there was a idea, I guess, paradigm in psychology called modularity, which had this idea that the brain had these different modules, which were almost like phrenological little blobs or areas that had very specific and narrow functions. And a lot of these modules were super simple, and it's sort of like some particular kind of information came into them. They performed some sort of operation or processing thing and pushed something out. And they were actually kind of dumb and blind to what they were getting. It's just like anything that's a visual stimulus, has some face-like features, I'm going to sort of respond strongly to that, call it a face, and send it to other areas. If something comes in and it doesn't have face-like features, I'm not going to do anything. So there's this sort of modular view that all these different brain areas have very specific functions. This is kind of a contrasting view that's saying, no, actually, like lots of the brain is involved in lots of things. We don't necessarily know how, we don't know what it means, but there seems to be a lot more going on than just one little blob of activation that's solely responsible or systematically related to face processing. So flash forward about 7 years, so this was originally shown and published in 2008, Kriegeskordy and colleagues came up with this idea of representational similarity analysis. And this sort of builds on that multivariate approach, but it also links back to, before the break, I talked about the condition-rich and time-continuous designs. And so what they do here, fairly complicated, but conceptually, I think you can get it. You have a bunch of different stimuli. And so in this case, we're using as sort of a toy example just 4 different stimuli. So we've got 2 faces and 2 houses, male face, female face, and then like a classical house and a modern architecture kind of house. So it's condition rich in that it's sort of condition poor in the sense that you've got faces and houses. Condition rich in that the 2 faces are different from each other in certain ways, and the 2 houses are different from each other in certain ways. So we treat each stimulus as its own condition, and compute these. They call them dissimilarity matrices. For simplicity, we'll call them similarity matrices. And so you can compute how similar is each pair of stimuli on some particular property, like luminance or brightness or something like that, and come up with a correlation matrix. And so that's basically what you're showing here is, the 2 faces are most similar to each other. So we'll say blue is more similar, red, yellow is less similar. The 2 houses are obviously going to be quite similar to each other, and then the faces and houses are more different from each other. So you've got sort of a way of quantifying how similar or different each stimulus is from each other stimulus based on visual properties. Then separately, you map out the spatially distributed pattern of activation in the brain that you measured with fMRI for each individual stimulus, and you build a similar matrix. So now the matrix is how correlated is the spatially distributed pattern of activation for the male face versus the female face, and for the male face versus the modern house, and that sort of thing. So now you have 2 matrices, one that tells you the sort of visual, physical similarity between them, and the other that tells you the brain activation similarity between them. And then you look at how well those 2 matrices map onto each other. And basically it allows you to test different models of what a particular brain area might be doing. So if the brain area is sensitive to just sort of face versus house, then it should show that gross level of sensitivity. As you build this out, you can actually have much more rich sets of stimuli with different kinds of objects, and they vary in lots of different properties. And that's kind of what's shown over on the right side of the screen here, is they actually applied a whole bunch of different models to just the visual stimuli, including some like deep neural network models of how primary visual cortex works and how other areas of the brain might work. Some simple sorts of filters that you might find in Photoshop. So passing like low pass and high pass spatial filters, different luminance filters, different sort of color filters, and so on. And then you can get all these different similarity matrices based on how similar all of your images are in color or in form and this sort of thing. And then look at how each of those relates to the spatially distributed pattern of activation in different brain areas. So separately for like fusiform areas versus other parts of the temporal lobe or whatever, and use that to say, okay, this is actually a very good model of how the fusiform face area processes information. Whereas this other model is a much better model of how primary visual cortex processes information. So it's a very nice way of looking and sort of exploiting the very rich spatial resolution of fMRI, which you lose if you're just sort of looking at blobs and thinking about modules, and relating it to fairly sophisticated models of how the visual system works or how the brain might work and so on. So you can start coming up with very computationally explicit models of how things might be processed and test them against brain activation. And so this has become huge, like at Cognitive Neuroscience Society meeting last year, which is the big cognitive neuroscience conference. I would say like the vast majority of the main central talks and the sort of hot topics sorts of things, we're using this in different ways and looking at how we can relate some of the very complex models that are being developed, like by Google for automated image recognition, if those actually reflect how the brain processes information. And it seems a lot of the time they actually do line up quite nicely. So I think it's a very important direction. So we're pretty much out of time. And of course, I'm not done. Never fails. So I think I will leave it for now, and I'll pack the information in. It's in the reading, obviously. I'll figure out the structural MRI chapter, which is next. I can spend a little less time on. So definitely read that for, I guess, after the break, as per the syllabus. But the first part of that lecture, I'll just finish up the stuff and then jump into the structural. All right. And then Wednesday we'll be back over there, and I'll have more EEG data sets for you guys to work with. It won't be your own data sets yet, but there will be more, and it'll be just practicing basically all the stuff that I showed you last week. All right, see you then.
We did not.
Okay.
Hey, yeah.
Because we're using faces in our study,
Uh-huh.
and I asked even if we have some faces from across or some databases that we could use.
Yeah.
I do have faces from the NIMSTIM database, but I got that for my thesis. I'm not sure how I can use them like because I mean they look the same, but like if I have the rights to use them now.
Right.
Stuff.
Yeah. I mean, it's probably safe, but if you email me, excuse me, if you send me an email, I can send you some stimuli that we've used in previous studies. Yeah. If you're just looking for like anything special or just faces.
Yeah, angry and neutral faces, like both female and male.
So anyway, so
Okay.
I remade this.
Send me an email and I'll see. I'm not sure if I have that, but those should be pretty available.
Also,
Like if you
like
start
you're
searching on the web, remember these
saying,
databases
about having like
are readily available.
because
Should be able to just
most of
access.
our experiments.
Sometimes you have to have like an actual prof or faculty member request them. So if you find one,
it's like
you can do that.
have more than
But start by sending me
goes
an
by
email.
going.
I'll see what I can do.
Yeah. So our ongoing stimulus changes. Oh, that was the instructions stimulus. I'll give you an image out of that. This is where I was trying to code in like the scoring, but I don't know yet how to do that. So I'm going to talk to you with 7 days.
So Liam was determined.
No, he said Google it, and which is totally fair. But like when I was like on the cycloplex forums, everything I found was like based on conditional loops and sending like if you have a right answer, using code to send someone down a different pathway, not having a score counter. This was super simple. It's just left right based storing the answer with like the correct answer that we have here. The stimuli, how long it's on for, the color. This actually threw me and Liz off like right after break because I've got it drawing from the color right here. It was white, gray, and black, but it defaults to a gray screen. So the gray arrows just weren't showing up. They was just showing up as like things. So we had to change it to blue. So I'm not sure if we're going to actually be able to draw it from the same color palette. You know what I mean? Like we were talking about different gradients of color, and I'm not sure if we can do that. But actually, here's someone we could ask.
Run away.
Bye. Look at the time. What's up?
With our EEG test, it's based on effectively seeing these and picking the opposite direction. So these arrows and picking the opposite direction.
Okay.
And having it scored a specific way. And one of the things that we were trying to do, just to make sure that everything was consistent, was have everything just be like drawn from the same color palette. So it was just using the same stimuli, white, gray, black. So there was like very little that would, I don't know, be a confounding variable potentially. And then when I was trying to do that in here, when it draws gray, it makes it as the exact same background screen.
Right.
So is it going to be an issue if we have like our 3 colors as white, blue, and black?
Why are you having 3 different colors?
Because
They're different.
yeah.
So your
Okay.
error has different consequences, basically.
Right.
And also the black is a different one?
No, I don't think we were going to. I mean, we can talk about that again, but from like just the conversation.
It's inside.
it seems- yeah. Anyway, so like when we go into here.
And maybe we only need to.
No, because we want to have,
The higher points.
yeah, you don't have that.
So it starts here. I changed it to press left or right to start because as soon as you start it, it kicks right off into the experiment.
The instructions.
Yeah,
So you're pointing the same way or the opposite way?
opposite way.
I feel like I
Of
did
the
this
middle
entire
arrow.
thing wrong.
So it's like a Stroop flanker.
Mhm. Yeah, exactly.
I would be
Okay.
going those things.
Oh my god, yeah.
I mean, in principle, it shouldn't matter what color they are.
Okay.
You might want to go with like red, green, blue, so they're all colors instead of one being a color.
But
'Cause
like
blue
a
might sort
deep
of jump
red,
out.
a normal red, and then a light
Like,
red.
or like we could do exactly that with the blues.
Yeah,
Yeah.
I mean, then you have luminance differences
Okay.
between the three. So it's
So-
better to actually use red, green, and blue of equal luminance.
Interesting.
Um,
Is there any way to like set that up in here? Because all we're using is like CyclePy's like draw from this color category in Excel.
Mhm. How do you specify the colors? Is it
So in color, I just hit draw from color from the Excel spreadsheet. Set with every repeat.
Oh, I see. So it probably has a few ways of specifying color. Like in computers, there's a number of ways. So RGB is where you specify the levels from 1 to 255 of red, green, and blue, and the combination gives you whatever color you get, right?
Yeah.
Another one is the hex that they use in web coding, which is like a hashtag and then 6 characters, which is actually a variant of RGB. When you use those, you can be much more specific. So if you, like in the Mac, color picker, so one of these, so here you can set them, find a color, or even you can go here, pick some color you want, and then go back here, and that gives you the RGB values, then the X values.
But then how would we get that encoded into like
Could you-
column C and then have CyclePy draw?
So it should, I'm going to guess that it will just read them, but that would just be something to check in the documentation.
Okay.
But if you just have like copy this and make sure there's a hashtag in front of it. So instead of red, it would be hashtag 0080FF.
Oh, okay.
But what you can do here is there are more sophisticated ways, but this is probably the easiest one. Pick here, actually, yeah. So here you can specify the brightness and the saturation and change the hue. So basically
only-
that means that
You're only changing the hue.
the hue is changing and the saturation and brightness are the same. And so you can find your colors here and jump back to RGB and pull up that code. and paste it in there.
Okay.
Yeah.
So cool, actually.
And when we're doing something like that, it's the brightness and the saturation we want to keep the same, not so much the color.
Yeah, and then vary the hue.
Yeah, hue. Thank you. All right. Anyways, so that's
Sweet. So the only thing we need now is the score counter.
yeah. And like I said, I'm going to see if I tend to